I'd like to thank everyone for taking time out of their days to join me on this webinar. I think what I have in store for you is a very interesting story about comparing two cleanup formats for QueChERS cleanup technique in the food analysis industry. So going over the outline for today's webinar, we're going to briefly overview pesticide monitoring in the food safety industry and what that entails. Then we'll step into the actual QueChERS technique, which is a very common technique for pesticide analysis in the industry. And then from there, we'll jump right into our evaluation of two cleanup formats. And we'll look at whether or not these cleanup formats can be considered QueChERS cleanups. And we'll also ask the question, is it more than a QueChERS cleanup? And from there, we'll revisit the conclusions in the summary and go beyond into the future of this technique. So pesticide monitoring for the food safety industry is an overwhelming task. If you look at the US FDA website, there's over 700 pesticides and industrial compounds that the US FDA monitors on an annual basis. And within that, there's a wide variety of sample matrices. And you can take these matrices and you can gather them into buckets and see that there are at least hundreds and even more different types of matrices that food testing labs will encounter. And this provides a unique challenge for these laboratories, because how are you able to analyze a wide variety of pesticides and compounds in such a wide variety of sample matrices? Another thing that's interesting to note from the US FDA website is you can look at the ranges of tolerance levels. Now, the tolerance level for any given pesticide or commodity is an action level, and where the EPA would take a regulatory action if it hits at that level or above that level. And you can see the distribution of tolerance levels in parts per million for the variety of commodities and pesticides range from anywhere from sub part per million to 1,000 part per million. But most of the pesticide tolerance levels range from 0.01 to 1 part per million. So as a reference point, I put a star right where our study spike level is at, which is 0.12 part per million. So we talked about the several different types of pesticides that are monitored in the industry and the different matrices. But when we look a little bit closer to at the different matrices that are monitored in the food and ag industry, you can kind of simplify it just by breaking it down in its most simple form. So the first form that I'd like to break it down is the sample type. Is it a solid or liquid? The goal of any extraction protocol for testing pesticides is to actually extract the target analytes from the sample. Whether they're in a solid sample or a liquid sample, you want to get those target analytes out of the sample. From there, I like to look at the sample composition. Samples can have a variety of different components inside of them, whether it's fats, carbohydrates, proteins. The goal of any analytical lab testing for small molecules such as pesticides is to remove those unwanted components and retain the target analytes in their extract for good quantification using their analytical equipment. And if you look at the triangle to the right, you can see AOAC food matrix triangle is a ranking system that allows laboratories to rank their sample composition. And it can range in percent fat, percent carbs, percent proteins, and anything in between. From there, once you've removed the unwanted components from your sample and retain the target analytes, the goal is to analyze the suite of pesticides that you're looking for. This is typically done using liquid chromatography or gas chromatography. So we talked about the different sample matrices, the components within, and the goals of any analytical lab trying to measure these pesticides in these samples. But what about the technique to do this? Well, QueChERS technique is a very common technique that's used for pesticide analysis. It's an acronym that stands for quick, easy, cheap, effective, rugged, and safe. And it is very much so all of those things. And this is why it's become very popular in many food testing laboratories and is even recognized in AOAC methodology as well as the European Committee for Standardization. So it's used worldwide as a standard technique. And the nice thing about this technique is it's very basic and it allows to simplify the extraction of such complex and wide variety of matrices. And in its simplest form, it has two steps. Step one is the solvent extraction, which is typically in acetonitrile. And as you can see from the picture below, we have spinach unhomogenized in a tube. And then after it goes through the step one homogenization and extraction, you sort of get a layering as seen in the picture to the right. So once you have that layering, you're able to take the sample from the top, the most darkest green layer in the picture, and transfer that into the cleanup step, which is step two. And from there, you're going to remove all the unwanted matrix components and leave the analytes of interest behind. So let's look at this extraction overview. Step one. So again, the targets can be in the liquid or it can be in a solid. And so you really have to understand what your sample matrix is or what your matrices will be that you will encounter in the lab and how you will extract the analytes of interest out of that sample. But the goal is the same either way. The goal is to get the samples extracted and the analytes of interest into an extraction solvent so that they can be further cleaned up for analysis. For our study, we're going to focus primarily on solid matrices. However, QueChERS technique is also applied commonly to liquid samples. So now that we understand the overview of the extraction, what is actually happening inside? What's the chemistry that's going on? Well, if you see from the images in the middle of the screen, you have your sample, and when you homogenize it, you will have a homogenate that has your fats, lipids, any of your unwanted components all homogenized with your actual analytes of interest and even internal standards that you use to basically understand how well your extraction chemistry is running throughout the whole protocol. Now, once you add your extraction solvent, you will also add these extraction salts. And these salts, depending on what method you're following, whether it's AOAC or the European Commission, will have a different range of different materials in them. Magnesium sulfate, which is used for removing any excess water in the sample. And it also will have these buffers, sodium acetate, sodium citrate sesquiterhydrate, and sodium chloride. And these buffer the sample to push the pH-dependent pesticides into the acetonitrile layer, which is, again, on the top. So what you'll happen through this step one extraction procedure is you'll extract the analytes of interest into the acetonitrile layer, but you'll also push the unwanted components out and the salt out of the acetonitrile and the water out of the acetonitrile layer with the salt. However, it's not a perfect extraction, and many of the fats and lipids and other compounds that you don't want do get retained in the acetonitrile layer. So from there, you need to clean up the acetonitrile layer that contains your analytes of interest, but also will contain your compounds that you do not want. And this is done with solid phase extraction, or SPE. Now, for traditional QueChERS protocol, it uses a technique called dispersive SPE, or dSPE. And the goal of the dispersive SPE is to put your extract in a tube with your SPE media and shake it up until all or most of the unwanted components will migrate into the sorbent layer at the bottom. And then at the top will be your extract with the analytes of interest, and hopefully none of them were retained into the sorbent layer below. Hopefully all of them migrated, and you'll be left with a clean extract that has your analytes of interest. Now, this can also be done another way. This could be done with column SPE or CSPE. And the nice thing about column SPE is there's actually different techniques you can use to isolate your analytes of interest. Now, the one that's most similar to the dSPE is what we call pass-through CSPE. And what you do for this is you pass your sample extract through the column that contains your SPE sorbent. And what will happen is the SPE sorbent will capture the unwanted components, and the extract that passes through is going to contain your analytes of interest. And it can be then collected immediately after being passed through, which is a parameter that we'll talk about later. And then on the other side, there's also what we call retention, wash, and elute. Now, for retention, wash, and elute, this allows you to get everything absorbed to the SPE sorbent, including the unwanted components, but then you can wash the unwanted components off the SPE sorbent and retain the analytes of interest. Once you wash them off, then you follow it up with the final elution to push the analytes of interest off. This typically leaves you with the highest cleanliness of extract. However, it's a little bit more cumbersome of a workflow. So for our study, we compared pass-through CSPE to dispersive dSPE, which is the traditional technique that is used for QueChERS cleanup. So now that we know what the format is and what the overall parameters are for performing this procedure, let's talk about the cleanup chemistry. Now, this is different from the extraction chemistry in step one. Step two is cleaning up, so removing any additional water that may have made it through the initial step one extraction. There will be magnesium sulfate that's in the tube that does remove that water. Now, one thing that's common between AOAC and EN methods is that they all use the same media. These medias are used at different ratios, but for the most part, they're all the same. And you can see PSA, primary secondary amine, is a SPE media that's really good at removing any carbohydrates or sugars that are in the sample. C18 is a hydrophobic sorbent that is very good at removing fats and lipids that might be in the sample. And then GCB stands for graphitized carbon black, and that's really good at removing chlorophyll and any pigments in the sample. And as you can see, for each method, AOAC or the European method, they have a cleanup sorbent that's used for general fruits and vegetables, more waxier fruits and vegetables such as oranges, and then pigmented or highly pigmented fruits and vegetables such as spinach. And the ratios of PSA to C18 to GCB will all relate to what the sorbent is supposed to be used for and what type of sample matrix it is. So we use this ratio to create CSPE columns that mimic the exact ratio that's used in the AOAC and European methods. And then from there, we extracted samples following both the AOAC and European Step 1 protocols. And then the extracts produced from those protocols were then spiked with a mix of 86 pesticides. Now, these pesticides represented a range of compounds that are encountered in multiple regions worldwide. So a pretty diverse range of pesticides. So once we spiked our extracts for both AOAC and EN, we then performed a cleanup. And we did the cleanup following the dSPE and CSPE technique that is equivalent to both the AOAC and EN procedure. And then we analyze, in this case, by GCMS. From there, we compare the cleanup performance, precision, and accuracy for both techniques. And we even do get a nice comparison of EN versus AOAC, which was something I did not foresee, but very interesting. So if you take a look at the recoveries for the AOAC pesticides, our first sample matrix that we looked at were oats. And we used the AOAC general cleanup sorbent, which has PSA and magnesium sulfate at a ratio of 1 to 3. Now, the blue bar represent the CSPE cleanup, and the orange bar represent the results from the dSPE cleanup, which is the traditional QueChERS cleanup. And they're all spiked at 120 nanograms per mil, as stated earlier, and these are 5 replicate spikes. Now, it's very difficult to see in this data. So you'll notice later on that I will actually bucket these results so you can have a better comparison. Now going to the European or the EN protocol in chemistry, you can see that the sorbent ratio for the general cleanup for EN is a little different. It's a 1 to 6 instead of a 1 to 3. And the results also seem a little different. But again, in the blue bars, we have the CSPE cleanups, and in the orange bars, we have the dSPE cleanups. Now, it's hard because there's 86 different pesticides that we're quantifying here. So what I found most interesting and easiest to compare is when you were able to bucket the actual data points. Now, here are the 2 data points that I just showed you, 2 charts for AOAC and EN, but now these data points are bucketed. And you can see this XY plot for EN. Any compounds that were greater than 130% recovery, I would bucket them. None showed up for the EN protocol. Now here's the sweet spot. This is where most people want to be in for their results. They want to have their recoveries anywhere from 70 to 130%. That's a good range of recoveries. And if you can see, most of the CSPE analytes fall within that range, as well as for the dSPE. From there, less than 70% recovery, which isn't as good, but at least it's quantifiable. You can see where the results lie for that. And then what's glaring is that the pesticides are the analytes of interest that aren't quantified. And you can see for dSPE, 20 of those were not quantifiable for 5 replicate spikes, whereas for the CSPE, only 2 of the 86 pesticides. And then over here, we have the same data set, but for the AOAC protocol comparing CSPE and dSPE. And it's pretty much the same story, although it seems to be a better overall procedure, the EN procedure for oats, at least, when compared to AOAC. But in both cases, the CSPE seemed to outperform the recoveries, the accuracy of the dSPE cleanup technique. So sticking with the EN, let's take a look at the RSD for oats. And I focused on the EN because the EN had better recoveries compared to AOAC for both CSPE and dSPE. So I thought it would be more interesting to look at the EN. And as you can see, for CSPE, most of the analytes, and even in dSPE, fell below 10% RSD, which is excellent. This is where you want to be at, 10% or less. From there, less of the analytes fell between 10 to 20, and then the same goes for greater than 20%, which is worse. But again, there were less compounds that were quantifiable for dSPE versus CSPE, and this is at a tolerance level, spike level. So that's not very good, and we are already starting to see the benefits of going with the CSPE. We're packing the same chemistry, sorbent chemistry, in a column and pushing it through the column, and it just seems to work better. So why is it working better? Well, it really provides a cleaner extract. It's almost as simple as that. If you compare the total ion chromatograms, or the TICs, for the standard just spiked and injected, this is just basically a straight standard and solvent. This is the CSPE after cleanup. And if you look at this baseline, it's definitely a little more shakier than the standard. But if you compare it to the dSPE baseline, you can see this is all the junk, the unwanted components from the sample matrix that you don't want and you want removed, so you get these nice sharp peaks that are seen in the actual standard. So this is primarily the reason why we are getting better recoveries and more consistent results. And what's interesting is, typically if you remove the unwanted components, you risk removing the analytes of interest and losing those. But we weren't seeing that, and so we are still getting great recoveries and cleaner extract, which is a win-win. So if we take a look at a single ion, you can see for penthiopyrad, the CSPE, you have a nice symmetrical peak, very little tailing, just really, really nice. Anybody doing chromatography really would love to see that. Now for dSPE, the same sim window, you could see you got peak splitting here. It's broad. It's just not the greatest chromatography. So this is really the significant thing that we are noticing between a column-based cleanup versus a dSPE cleanup. And again, remember, these are the same ratios of SPE medias, just one's packed in a column and one's loosely placed into a tube. So let's take a look at another matrix. So we see this is apple, and you can see the AOAC pesticide recoveries for apple. And again, we used the general AOAC cleanup ratio, PSA, and magnesium sulfate. And you can see overall, these analytes for the CSP seem kind of low. Well, actually, what we noticed, and we will work towards placing this in another webinar, because it just was too much data to actually add it to this webinar. But what we notice is we have an opportunity with CSPE to do a follow-up elution. And when you do that, you can actually push these recoveries up significantly. And so not only are you getting more analytes to quantify, but you can actually push these up. So we did a side-by-side comparison of this, and we actually improved the CSPE data with that. But taking a look at the EN pesticide recoveries from apple, you can see that the CSP and the dSPE are very similar in recoveries. However, there's some of these analytes that aren't being quantified by a dSPE that you can only really see when you bucket the data. And so I'm as you will find out, a big fan of bucketing data. So let's look at the bucketed data. So we have the Apple matrix here now, and we're looking at the same two charts, but we're bucketing the data as we did before. And what you'll notice again is in terms of the analytes not being able to be quantified, dSPE definitely had more. Wasn't as bad as with the oats, but it was definitely again underperforming compared to the CSPE. Overall, I think they both did very good, but definitely saw better results in the CSPE and better results overall in the EN technique, European technique. So again, looking at the RSD values for the EN, since that outperformed the AOAC technique, again, we're seeing some good distribution of the RSDs, most of them falling in the range that I would say excellent, less than 10%, with some falling between 10 to 20, and not as many falling over 20%. Again, there were more analytes that weren't quantified by dSPE compared to CSPE. Overall, the CSPE again outperformed the dSPE. And so it tells the same story. I mean, no matter what matrix we're looking at, we're seeing the same story. We're getting a cleaner extract from the actual cleanup procedure using CSPE versus dSPE. And in this case, it's again in that same region, very similar region. And this is why the data we're able to quantify more analytes compared to the dSPE. And let's take a close look at fluxapyroxad, and you can again see this is just for the dSPE. This is just not chromatography anybody really likes to see. But wow, for that CSPE, it just looks so much better. So without going through each matrix evaluated, I'll just kind of give a summary of which ones were also evaluated. We did oranges with the waxy cleanup technique, broccoli with the pigmented, and spinach with the highly pigmented. I did add a picture of the spinach. As you can see, this was before the cleanup. And then this was the dSPE cleanup. So this is after cleanup. It's a little lighter, but you can see with the CSPE cleanup, much cleaner extract. And this is why you're getting just sims like this, where you just have all that junk for the dSPE, all those unwanted components, they're being removed, and you have a nice symmetrical peak with little tailing. So this data will be available after the webinar, and I think we've shown in these data sets that we're able to give the accuracy and precision equivalent, and maybe in some cases much better than the dSPE technique. So from here, I really like to ask the question, is it QueChERS? And I think we've already kind of demonstrated that it's effective, it's rugged, so you're getting effective results. It's reliable sample to sample, and it's safe. You're actually exposed to less solvent doing it this way than you are doing the dSPE way. But what about the three other parameters? Is it quick? Is it easy? And is it cheap? And that's really what makes, outside of the chemistry of the QueChERS technique, it stands for quick, easy, cheap, effective, rugged, and safe. So you have to really hit all those points to call it QueChERS. So if we look at quick and easy, for dSPE, if you did a manual shake of your samples for the cleanup, and this is 48 samples being cleaned up, and the orange bars are for dSPE. If you did a manual shake, which we did two tubes per hand, so 4 samples at a time, manually shaking it by hand, which isn't fun. But it's actually relatively quick. It takes under 2 hours to do, and that's actually not bad for 48 samples. Now, some labs are doing, 48 plus samples a day, so that could get pretty tiresome. There are automated shakers. You could use for instance, the Biotage Lysera to do some of that shaking for you. However, it's not easy to completely automate it. There are ways to do it, but it gets very expensive and very complicated very quickly. So I put automated with an asterisk because it's hard to automate, and I'll get into that a little bit more in a couple of slides. Excuse me. But when you compare this to the CSPE technique, um, funny thing is the manual is very easy. The manual we use a positive pressure system, the Pressure Plus, and it is very simple to do. Elutes directly into the analytical vial, so that saves time right there. You save a transfer step. So 19 minutes for 48 cleanups. That's pretty impressive. And for automated, it actually takes a little longer, but it's automated. So you have that. And overall, very quick. Now, cheap. And cheap is, when you look at cheap, you kind of have to look at what's the sorbent that's used in the QueChERS technique. Now, the traditional QueChERS dSPE uses about 1 to 2 grams of sorbent in the tube. And that's the bulk of your cost right there. I mean, you have the cost of the tube, the cap, but the bulk of your cost is that sorbent right there. So if you look at, a 15 mil column SPE, that can pack anywhere from 1 to 5 grams of sorbent. You don't want to pack too little, or else it doesn't distribute evenly across the actual tube. But that will actually be more expensive, and that's not the column format that we used here. 6 mil gets a little bit similar to the price of the 15 mil, and then your sorbent range goes from 0.1 to 2 grams. But for us, we decided that it was optimal to downsize or miniaturize the technique and go even lower in volume, and that actually is going to be less expensive than a traditional technique. From there, you could even go lower in size. And what's nice about SPE, and I'm sort of spoiling some of the slides upcoming, is it's very easy to miniaturize, especially on a positive pressure system. So let's answer the question: Is it QueChERS? I think after all the data and evaluating how quick it is and even the cost, giving a relative evaluation of the cost, I think it does fit the QueChERS. So I really believe that CSPE, this QueChERS CSPE, can be a good alternative to the dSPE. Well, from there, I guess the next question would be, is it more than QueChERS? And I thought of how it could be more than QueChERS and what would make it more than QueChERS. And, taking some intel I have from traveling to different labs in North America, and what I hear from different end users and that are doing QueChERS and performing it. And some of the parameters that stand out is it easily automated? I think that's a big deal for people. Can you easily automate that technique? Are you able to do alternative SPE chemistries? That's another question I wanted to answer. Is it open to modern prep techniques? And then finally, is there miniaturization potential? Can we scale down? The more you scale down your operation, the more throughput, the more samples in, the more samples out you get, less solvent. So miniaturization is a huge benefit to labs. So easily automated. And this is where I put the asterisk before. QueChERS, the traditional dispersive SPE, is not easily automated, and that's due to the fact that you have to not only transfer the extract to the tube, then you have to cap it, and then shake it, and then so shaking it, you could have an automated shaker, but then you have to transfer it from the shaker to a centrifuge, because then you got to centrifuge it down, and then you have to uncap it, and then you have to transfer it. So you really have extra steps, and it's really difficult to automate all those steps. But the nice thing about the CSPE process is you can have the system transfer the supernatant, the extract to the SPE column. It'll push it through, the system will push it through the column, and it will actually collect it all in the same step. And so you minimize the steps, and it's much easier to automate. And that's one of the biggest benefits outside of um, the actual data benefits. So the second question is it is there alternative SPE chemistries available? And I'm going back to the a slide a couple of slides ago, comparing the different mechanisms for SPE cleanups, and you can see that it's to have the retention wash elute be done via dSPE would be very difficult. It's much easier to do it on column SPE. So I think the column SPE is actually more adaptable to different SPE chemistries. And like I said before, you will get an even cleaner extract. This is used primarily for more targeted list of analytes. If you really know what your analytes chemistry are and they all follow the same unique chemistry, you can really hone in and get very targeted clean extracts with your analytes in them. So the nice thing about doing it this way for QueChERS is you have options for more advanced or alternative SPE chemistries. And then is it open to modern techniques? One of the things about QueChERS is it's there. It's QueChERS, it's great, it works, but what else can you do with it? And most sample prep for solid phase extraction is developed on cartridges or plates, and there's new techniques that go beyond, and some are even simpler than the solid phase extraction techniques. One being supported liquid extraction was a very, very simple technique, but is used by several labs for quick and effective cleanups. Dual mode extraction is another technique that can be used. Protein precipitation is a unique technique, and phospholipid depletion is also another technique. And these all are applicable on the same systems that process the QueChERS columns. So if you want to transition into a more modern technique that might be even simpler and might give you better results, then you have that option as well. Sorry about that. And then from there, the final question is there miniaturization potential? And with dispersive SPE, it would be difficult to miniaturize it beyond what's already being miniaturized. I think people will go as low as about 1 mil for dispersive. But what's nice about column SPE is there's new technology called micro plates. And these are not just 96 well plates, which have also been around for a while. These are actually micro plates, so they're packed in the actual tip. And this allows you to pack as little as 2 milligrams of sorbent, which allows you to elute volumes as low as 20 microliters, which would be extremely difficult to do in a dispersive way. So there's really endless potential for miniaturization using positive pressure processing and CSPE cleanups. So I think that it is more than QueChERS. It really allows you to expand your capabilities and try different chemistries that go beyond QueChERS. And really the reason why we developed this technique is to mimic what people were already doing, the same chemistry, but give them a mode, a means to transition into new techniques, alternative chemistries that use the same processing platform. So from there, I'll end with some conclusions and go a little bit beyond. first conclusion, of course, is uh, the CSPE for catchers, isolute CSPE for catchers, they are definitely an effective alternative to traditional dSPE. Uh, and that was proven here. They really produce clean extracts for a wide range of produce types. The results are very accurate and reproducible compared to the dSPE. There's elimination of manual transfer steps, which leads to higher throughput. And you also have options for both manual and easily automated processing. The second point I want to make is, like I said in the last slide, the CSPE, isolute CSPE for catchers, they provide a way to transition from the traditional technique, which is dSPE, to CSPE with minimal changes to chemistry. We did that on purpose. We wanted to mimic the dSPE sorbent ratio. Oh, excuse me, dSPE sorbent ratios with the CSPE, and that allows labs to make minimal changes to their chemistry and transition easily to the CSPE. And then it also allows them to use platforms like the Biotage Extra Hera or the Biotage Pressure Plus, which are positive pressure platforms. And these provide even more benefits beyond the dSPE, which is the fact that they can be easily automated. They're open to alternative SPE chemistries if you want. They have modern prep techniques such as SLE, dual mode extraction, protein precipitation, and there is great potential for miniaturization even further into the future as scaling down is just becoming more and more important for labs these days. And with that, I will thank you for joining me today. I'd like to acknowledge Lucy Lunn especially for all her hard work. She's the mastermind behind all this data and did a fantastic job and made it really easy for me to create a nice story out of her results. And also the rest of the team out in Cardiff, our R&D team for Biotage. I thank them all, and I thank you for joining, and I'll leave it open for questions.
Well, thank you, Evan, for your excellent presentation. We now have time for some questions. For those of you listening who have questions, now would be a very good time to send them in. Simply click on the Ask a Question button, type your question into the box, and click Submit. There will also be some time after the session has finished to type your questions in, so please do so, and they will be answered offline afterwards. So our first question is, can you utilize vacuum manifolds to perform the QueChERS CSPE cleanups?
Yes, that's a good one. Yeah, vacuum manifolds are very common. People typically already have them. They're low cost for SPE, so they actually can be used. We have protocols to help people use them. However, I will say the positive pressure does make a huge difference, and we will have more info on this. The nice thing about the positive pressure is you can really control, very, very finely control the speed at which your samples are processed through the column, which makes a difference in terms of cleanup and cleanliness. So when you break it down, the positive pressure manifold, the manual one, in terms of cost and investment, it is not expensive, and it is worth, it might be a little bit more than a manifold, but it is worth the extra investment.